Welcome to Suhail's Paramedical and Nursing Academy. Today I am going to explain about the cell cycle and cell division. So in the previous lecture I was explaining about the peritoneal dialysis, then I told that I will explain the genetics. Before entering into genetics, before entering into the genetics, we have to know about the cell cycle and cell division. When we are knowing the cell cycle and cell division, then only we can proceed towards the genetics, because cell cycle and cell division is the main concept. Actually I recorded this lecture in the previous day, but due to the technical issue in the camera it was not recording. So then I was thinking to record once more the same video. I was thinking to record once more, but due to some reasons I was not feeling well. So today I will explain about the cell cycle and cell division. Okay, so today what is the topic? Cell cycle and cell division. First of all, we should know what is the cell cycle and what is the cell division. So after understanding this lecture, I will remember one thing, that after you will study the genetics from me, you will understand that there is possibility, in case of some species there is possibility they are giving birth to the new ones because of mitosis by means of mother. There is no involvement of father, so only father gives birth to the only mother gives birth to the new one. So that is also the concept you will study, that is only from the genetics. So because for that purpose, for those things, mitosis is important. Today will be the mitosis, next time I will explain about the meiosis. So first is cell division. In this we will explain about the anaphase. The anaphase will be here mitosis. Okay, so first of all, what is the cell cycle? I will give a quick review from the board itself. What is written here? What is written here on the board? I will explain the same. After this, I will put the screenshot in the. There is my Instagram channel is there. So here is Paramedical Nursing is Instagram account. There also will be the same. It will be the same content. Will be on the Instagram also. I will put this. And one more thing is there. You can write with me whatever I am saying. You can write with me. First of all, we will start from the cell cycle. Actually, cell cycle in case of humans, cell cycle means what? The cell cycle is a whole process. Cell cycle is a process in which cell prepares for its division and finally undergo division and form two daughter cells. I am telling about the mitosis, but in case of meiosis, it will form four daughter cells. Okay, so two daughter cells will be formed from the cell. This whole process is called as cell cycle. So what are the phases in the cell cycle? There are two phases in the cell cycle. One phase is called as interphase. This interphase is also called as preparatory phase. This phase is also called as growth phase. Okay, and another phase is there. Another phase is called as mitotic phase. Another main phase is called as mitotic phase. In case of mitotic phase, remember, in case of mitotic phase, what will happen? That will be totally different. So mitotic phase, remember that in case of mitotic phase, there will be two phases. One is called as karyokinesis, another is called as cytokinesis. So in case of cell cycle, there are two phases. One is called as interphase, another is called as interphase, also called as growth phase, also called as preparatory phase. Another phase is called as mitotic phase. Mitotic phase is also called as mitotic phase. It is a dramatic phase. It is also called as a dramatic phase. This phase consists of two parts. This phase consists of two parts, two stages. One is called as karyokinesis, another is called as cytokinesis. Okay. But interphases consists of how many phases? G1 phase, S phase, and G2 phase. Remember. So actually, its cell cycle starts from G1, S, G2. Then karyokinesis. In karyokinesis, it starts from prophase, metaphase, anaphase, telophase. See, I have shown here. First phase will be the G1 phase, S phase, and G2 phase. Okay, G1, S, and G2 phase. These three phases are called as interphase. Collectively, these are called as interphase. Remember, this prophase, metaphase, anaphase, telophase is called as karyokinesis. Then there is cytokinesis. Both karyokinesis as well as cytokinesis is collectively called as mphase, mitotic phase. Okay. Then, so this was the interphase G1, S, and G2, and this was the mphase. Mphase means prophase, metaphase, anaphase, telophase, then cytokinesis. This is the stages in the cell cycle. So what will happen? I will explain stage by stage. Okay. So So what will happen? The cell is called as cell cycle in case of human is mostly 24 hours. Remember this, and 23 hours are consumed by the interphase. So interphase is the longest phase. Only 1 hour is consumed by the mitosis. Only 1 hour is consumed by the mitosis. So what will happen? Why it is called as interphase? Why it is called as growth phase? Why it is called as preparatory phase? Remember, it is called as growth phase because in this stage the growth of the cell will occur. The growth of the cell will occur. The cell will increase its cytoplasm. The cell will increase the DNA content. The cell will double its DNA content. The cell will double its centrioles. Okay. Also, the cell will increase its organelle number, cell organelle number, and in this that is why it is called as growth phase. It is also called as preparatory phase. Why preparatory phase? Because in interphase, in this phase of cell cycle, the cell will prepare for the division. Division will not occur in this phase, but cell will prepare for the division. That is why it is called as preparatory phase. Another phase is mitotic phase. So that will come after that. But first I will start from here. Interphase. Why it is called interphase? Why it is called growth phase? Why it is called as preparatory phase? I have already told. But here remember G1 phase, S phase and G2 phase. It will start from actually G1 phase. Okay, interphase will start from G1 phase. In case of G1 phase, the G1 phase, what is the G1 phase? G1 phase is the metabolically active stage. In this stage, the cell is metabolically active. Metabolically active means there will be metabolism as well as catabolism. The metabolism and catabolism will occur inside the cell. Okay, the anabolism and catabolism will occur inside the cell. That is why it is metabolically active. And in this state of cell will grow. The cell will increase its cytoplasm. The cell will increase its organelle. Cell will increase its organelle number. Here I have written organelle number. The cell will continuously grow. It will increase its cytoplasm. The ribosomes will be also increased. If there are 5, 10 ribosomes, these ribosomes will be also increased. Okay, and cell will increase its proteins required for the division in the future. The cell will increase its RNA. The cell will be transcriptionally. Remember, the cell will be translationally active. So transcriptionally the cell will be active. Okay, this is called as gap phase first. It is called as G1 phase, also called as gap phase first. Another phase is called as S phase. S phase is also called as synthetic phase. S phase is also called as synthetic phase. Remember why it is called S phase, why it is called as synthetic phase. In S phase or synthetic phase, there will be the synthesis of DNA. There will be the duplication of centrioles. That is why it is called as S phase. It is called as synthetic phase because synthesis of DNA occurs in this stage. So remember, DNA replication occurs in the S phase, centriole duplication occurs in the S phase as well as. Remember, what is the point that is important in this stage? What is the point that is important? That cell, in case of S phase, DNA replication occurs. The DNA replication means the cell will increase its DNA content into double. So content of DNA will be double. But after S phase, the number of chromosomes, the number of DNA will be the same. Remember, so that is why in this stage, from cell will increase the DNA content from 2C to 4C. Content will be increased. That is why it's called. That is why content will be increased from 2C to 4C. Okay, but the cell is in the 2N stage. It will remain in the 2N yet. Why it is the meaning of 2N? See, I will give explanation here. Cell. This means why it is called 2N and 2N. See, this is the father's chromosome. This is the mother's chromosome. This is father's chromosome. This is mother's chromosome. It was also having one copy. It was also having one copy. One copy of father, one copy of mother. Okay. Remember, father's copy will be also increased. Mother's copy also will be increased. But see, this father's copy is called N. This mother's copy is called M. So N and N means 2N. So there are two copies of both. There are two copies of chromosomes. One will be from the mother. One will be from the father. What happened here, that both father's as well as mother's copy has been replicated. So but it is in 2N stage. 2N means one from mother, one from father. But this N, mother's copy as well as father's copy has been increased. So father's copy and mother's copy increased and they will remain attached. Two father's copy are attached, they will act as the sister chromatids. Two mother's copy are attached, they will act as the sister chromatids. So hence the DNA number will be same. The number of chromosomes will be same, as well as there will be no increment in the content. There will be no increment in the in the variety. Variety means what? Mother's copy also increases one more time, father's copy increases one more time. So there is no net new any variety. Same copy has been increased, mother's copy, father's copy. So this is mother's copy, this is father's. So it is yet 2n. It is yet 2n. So 2n from 2n to n. That is why when anaphase will occur, these sister chromatids, I will tell later what will happen. So you will understand this much of point. That is 2 from 2c, it will go to 4c. DNA content will be increased, but number will be remain same. Number 2n to 2n, it will remain 2n to n. One number will remain the same, as well as father's and mother's copy will form one more copies. Okay, and those copies remain attached as a sister chromatids. That I will show you later what will happen. So then there is G2 phase. In case of G2 phase, further growth of the cell occur. The cell, if cell requires any more growth, the further growth will occur into G2, yeah, or we can say gap phase second. G2 phase or gap phase second, and spindle fibers will be also the polymerization of spindle fibers, the formation of spindle fibers from the centrioles will also occur in this gap phase second. Okay, then there is a phase that is called as mitotic phase. So this was the growth phase, interphase, preparatory. Now you can understand why it is called a growth phase, why it is called a preparatory phase. Okay. So now we will go towards the mitotic phase. Mitotic phase. Before I am going to tell about the mitotic phase, I will go towards the diagram. First, see, mitotic phase occurs in only 1 hour. That is why it is called as dramatic phase. Most dramatic phase. It is the very short phase. M-phase is the very short phase. It occurs in where? Where the mitosis occurs? Mitosis occurs in the somatic cells. The normal body cells, the body cells which are present in our body, except germ cells, those are called as somatic cells. It occurs in the somatic cells, as well as the same mitotic division occurs in case of primordial germ cell. Remember, the primordial germ cell, we all know that during the spermatogenesis, during the oogenesis, what will happen? That there will be a cell that is called a primordial germ cell. That primordial germ cell undergo mitosis. Then further it will go for meiosis. Then further the oogonium, or we can say spermatogonium will undergo for the meiosis. That is different. That is in case of reproduction. But they will first undergo mitosis. The first stage will be the mitosis, that is shown by the primordial germ cell. That is the gonads, the testes, the ovaries will show. Are having primordial germ cell, they will show mitosis first. Okay, that is why then DNA in case of interphase it will be less condensed. Less condensed DNA will be the hydrated form of DNA. It will not be visible in the cell. The less visible, the less condensed, less visible hydrated form of DNA which is present in the cell during the interphase is called as chromatin. This chromatin will be present in the interphase. After interphase, this chromatin will convert into chromosome. How? Because in case of mitotic phase, the chromosomes will undergo what? Condensation, dehydration, condensation, and the thickness of the chromosomes will increase. The chromosomes will shorten. The size of the chromosome will decrease. They will shorten. And what will happen? The condensation will occur. That the dehydration of the chromosomes will occur, and chromosomes will become visible. So, in case of interphase, DNA was in the invisible form, that is called as chromatin. But in case of mitotic phase, it will become more visible, more condensed. That is now it will be called as chromosome. Remember. So, in case of prophase, there is the first phase. In case of mitosis, there is karyokinesis. First phase is mitosis, prophase. In case of karyokinesis, there is prophase. First phase. In case of prophase, what will happen? The condensation of DNA will increase. The galactonemic coiling of chromosomes will occur. See, this is called as galactonemic coiling. What is galactonemic coiling? This is the one coiling. This is called as galactonemic coiling. But in case of meiosis, paranemic coiling will occur. That paranemic coiling is like this way. Okay, so that coiling is very less. That coiling is not so much coiled, but this DNA is very high coiled. That is why it is called as galactonemic coiling will occur in this state. Condensation of chromosome will occur. The chromosomes will condense in the prophase. Remember, I told condensation of chromosomes will occur in the prophase. So chromatin threads, it was present in the form of chromatin threads. Now it will separate from the chromatin reticulum and it will convert into chromosomes. Okay, and the centrioles will move opposite to moles. There was presence of centrioles. They will try to move. These are centrioles. They will move towards the opposite pole, towards the opposite poles of the cell. And the DNA was present in the form of monad. The DNA was present in the form of monad. It will be visible in the form of dyad. Dyad means what is dyad? Actually, see, this was the monad. Monad means only one. See, this is called a monad. Only one sister chromatid was visible. Only one sister chromatid was visible. After the prophase, in case of prophase, in late prophase, what will happen? One more sister chromatid will be visible from this DNA. That is called, it is called diad. It is called as diad. So only one was visible. But in case of what is called as prophase, late prophase, the diad will be visible. Another thing is nuclear membrane and disease. See, nuclear membrane and nucleolus will disappear. Nucleolus is present inside the cell. I have already explained in the cell structure. Nucleolus will disappear, as well as this. The nuclear membrane will also undergo fragmentation. The nuclear membrane will also disappear. Why there is occurrence of disappearance of nuclear membrane? Why nucleolus is going to be disappeared? The reason is clear. Because cell has to undergo division. The cell has to divide the chromosomes. The separation of sister chromatids should occur. The separation of sister chromatids at the kinetochore of the chromosomes, at the centromere of the chromosomes, the division of centromere will occur. When division of centromere will occur, for that we have to attach the spindle fibers. Spindle fibers will be formed from the centrioles, two centrioles from the opposite poles of the cell, two centrioles from the opposite poles of the cell. Remember, for that purpose, in cytoplasm, we have to remove the nuclear membrane, we have to remove the nucleolus, we have to remove the organelle, we have to disappear the organelle. Then only spindle fibers will attach on the chromosome on both sides. So, see, it was one form was already there. One form mother, one copy mothers, one copy fathers. Now one more copy has been increased, and those two copies are attached to each other. See, in case of metaphase, what will happen? Now it will enter towards the metaphase. In metaphase, the chromosomes will lie at the equator. The chromosomes will lie at the equator. They will lie at the equator. The spindle fibers will get formed. The spindle fibers will get formed. Those spindle fibers will attach on both sides of the chromosome. Remember this point. In case of metaphase, spindle fibers will attach on both sides of the chromosome. On both sides of the homologous chromosome. Remember, I will tell later what is homologous chromosome. On both sides of the homologous chromosomes, and there what will happen? So they are analysis. In case of metaphase, chromosomes will arrange at the equator. Spindle fibers attach both sides of chromosomes at kinetochore. Maximum condensation will occur in this stage. So DNA will be more condensed. We can karyotyping. That is we can easily, this DNA will be easily visible in case of metaphase. These in the form of chromosomes. Okay. So first they arranged in the equator. Then spindle fibers attached. Now spindle fibers are attached and they are present at the equator. What will happen? See, I will show diagrammatically. See here. See here. What is this? See, these are called as sister. This is called as chromosome. This is called as homologous chromosome. This is also called as homologous chromosome. This is the copy of father. This is the copy of mother. This is the copy of mother. This is the copy of father. This is homologous chromosome. This is also homologous chromosome. These two are called as sister chromatids. These two are called as sister chromatids. These two are also called as sister chromatids. But this and this is called as non-sister chromatids. These two are sister chromatids because this was a copy of father. One more same copy has been increased. That is why they are each other sister-like. They are like a sister. So they are sister chromosomes each other. Their DNA is same. That is why one more copy of father has been increased here. Okay, father's copy, one more. So this is homologous chromosomes. This is homologous chromosomes. These are called as sister chromatids. And this is the copy of mother. This is the copy of father. Okay, and these spindle fibers will attach here. That's called as kinetochore of the chromosome. Here also it will attach. That is kinetochore of the chromosome. But in case of metaphase, in case of mitosis, what will happen? They will arrange like this way. They will arrange like this way. See here. They will arrange like this way. In case of mitosis, they will arrange here. So on the both sides, here also, spindle fibers will attach on the both sides. Here also it will attach on the both sides. See, both I told in case of metaphase, chromosomes arranged at the equator. After chromosomes arranged at the equator, what will happen? That spindle fibers will attach on both sides of the chromosome. Spindle fibers will attach on both sides of the chromosome, and the maximum condensed DNA will be visible only in the metaphase. Okay. Then in case of anaphase, the next phase will be anaphase. In anaphase, these spindle fibers will undergo depolymerization. Depolymerization means these there will be, in case of spindle fibers will short. The spindle fibers will shorten. The spindle fibers will shorten, and what will happen? What will happen that the two sister chromatids of the homologous chromosomes will separate. When the two sister chromatids of the homologous chromosomes will separate, they will move towards the opposite pole. That is called as anaphase. Okay. So remember that in case of genetics, we will study about the genomatic mutation. The genomatic mutation occurs when there is non-disjunction of the homologous chromosomes. So non-disjunction means they will not separate. When any mutation came, because of that, the two sister chromatids are not separating. When they are not separating, they will enter one side, and one side, that one sister chromatid will be less. So that will create a mutation. I will tell later what are the different mutations that will occur. So they may occur in the anaphase. Then there is telophase. Telophase, now DNA has been separated. Now in telophase, what will happen? Telophase is the same what is there in the interphase, except S phase and synthetic phase. Actually, what will happen in the telophase, I will tell once more later. What is actually telophase? See, you can write in the same. In telophase, that there was cell organelles which was already disappeared in the metaphase. In telophase, these cell organelles will become visible. The cell organelles will become visible. The nucleolus will become visible. The nuclear membrane will become visible in the telophase. And the chromosome which is present in the condensed stage will now decondense in the telophase. Decondense means it will be like a chromatin reticulum. Chromatin reticulum means in a decondensed stage there is a chromosome. One chromosome is there. That chromosome is called as chromosome number 17. In our genome, the chromosome number 17 has a gene. That gene is called as P53 gene. The P53 gene undergo transcription, then translation. It forms a protein that is called as P53 protein. The P53 protein is called as the watchman of genome. It is called as the watchman of genome. Why it is called as the watchman of genome? It will act as a watchman. Remember, it will act as a watchman. If there is any kind of mutation, it will be the watchman. It will be watching everyone. Whatever is going in during the cell cycle, it will prevent any kind of mutation. So remember P53 protein. Another is cyclins and CDK kinase. Cyclins and CDK kinase are some proteins which will also prevent any kind of mutation. They will also help in running the cell cycle and cell division in a normal way. They will prevent any kind of mutation. And these cyclins and CDK kinases will act at some checkpoints. Those checkpoints will be between these checkpoints will be between G1 and S phase and between S phase and G2 phase. Remember, so that if any kind of mutation, if the cell is not ready at the G1 phase, then the cell will not enter into S phase. If the cell has replicated the DNA in the S phase, if any mutation came, it will not go towards the G2 phase. So this will be done by the cyclins and CDK kinase. They will also act like a watchman. Okay, so this was all about the cell cycle and cell division. One more thing you should, once more, this was all about, I will give a quick review. Quick review will be there, just 2 minutes quick review. What will be quick review? This is cell cycle. Cell cycle has two parts. One is called as interphase, another is called as mitotic phase. Interphase consists of many stages. Interphase consists of G1 phase, S phase, G2 phase. Mitotic phase consists of prophase, metaphase, anaphase, telophase. In case of interphase, the G1 phase, what happens in the G1 phase? I already told you, there will be the growth of the cell. What happens in the S phase? DNA replication and central duplication. What happens in the G2 phase? Further growth occurs in the G2 phase. Then, now the cell is ready for the division. It will enter the mitotic phase. In mitotic phase, what will happen? There will be the prophase. In prophase, the cell organelles will disappear. The nucleolus, the nuclear membrane, the nucleolus and nuclear membrane all will disappear because what will happen? The polymerization of the spindle fibers will occur in the prophase. The DNA which was present in the reticulum form in the interphase, now in case of M-phase, this DNA will be more condensed and it will be called as the chromosome. After prophase, it will enter into the metaphase. In metaphase, more condensation of DNA will occur and there will be the appearance of homologous chromosomes. Those homologous chromosomes, one homologous chromosome from father, one homologous chromosome from father. These two are, the homologous chromosome consists of two sister chromatids. Father's homologous chromosomes, homologous chromosome, mother's homologous chromosome. Both are having sister chromatids. Okay, remember that also. And in case of anaphase, the separation. So in case of metaphase, they will arrange at the equator. These sister chromatids, these chromosomes will, homologous chromosomes will arrange in the equator in such a way so that sister chromatids will attach on the both sides. And in case of anaphase, there will be separation of sister chromatids. And in case of telophase, what will happen? Once more, the cell will enter into the decondensed stage. That DNA will be decondensed. The nucleolus, the nuclear membrane will reappear, will get formed. The organelles which are required will get formed. So everything will come in the in telophase. The same will happen. The same will happen as like suppose in case of G1 phase, what was happening in case of prophase. Suppose in case of prophase, what was happening in prophase? Organelles was disappearing, nucleolus, nucleolus and nuclear membrane was disappearing. But in case of telophase, opposite will come. Opposite of the prophase will come in the telophase. I hope you will understood. If still you have any kind of doubt, I have already told that there is one. You can contact me. You can contact me on my channel on Instagram. That is called as suhailsparamedicalnursing. On that Instagram, you can message me there. I can solve the doubt. If you are not able to tell there, you can tell here on the video in the YouTube. And further, if you don't want to tell the doubt on the YouTube also, then you can contact me. I will explain the doubt on the next video. Thank you so much. Thanks a lot.